Thank you, all. So I hope you can hear me all right, since the speakers are in front of me and not behind me. So the point that I'm going to be focusing on today is a little bit of an overview of what the bioconjugation process is, followed by some discussion of the project management aspects of bioconjugation. So I'll give you a brief overview. You've already seen some slides on this topic, so I won't dwell too much on ADCs. And then I'll talk a little bit about the whole complexity of the process and how to manage it, and talk a little bit about what the difference might be between a small molecule and an ADC. We okay? So just a general overview in terms of when I talk about ADCs in this talk, I'm referring to any kind of bioconjugate. That would include synthetic vaccines or any type of conjugation or coupling between a small molecule and a protein. And one of the things that is general to these types of projects, which is also small molecules, are the considerations that go into it, what phase you're at, the number and status of analytical methods, the conjugation scale you're looking at, the sourcing of the antibody as well as your payload, and then your fill finish and stability studies, where those are being done. And over all of these, of course, are regulatory requirements. So you've seen some slides already about ADCs. The general concept is that you have a biomolecule that has a very specific targeting, selective binding, and this, in many cases, to tumors, can also be to other cells, long half-life in the system, non-toxic in circulation. However, the therapeutic effects, particularly related to cancer, are limited. You have cytotoxic agents, traditional chemotherapeutics, for example, highly toxic, but no specificity. And then the ADC, which is sort of the magic bullet, very specific, very high toxicity, and on-target delivery as the goal. A little bit more of a cartoon. Some of the issues that need to be considered that are part of the whole development of an ADC. You have the antibody. As we've heard in previous talks, these can be fragments, these can be any number of things. The ones on the market right now are IgG whole antibodies. High affinity, and they're usually associated with an overexpressed antigen on the target cell, tumor site in this case. You have a conjugation strategy that you have to think about. How is it being coupled? The linker is very important. Early history of this showed some failures in development where the linker selection was inappropriate. And then finally, of course, you have, again, the payload, the toxin linker, highly potent, and it has to, of course, be suitable for the indication that you're targeting. So the overall strategy, this is a little bit of a historical slide. Some of the developments that have been happening, IgG, simple immunoproteins, diabodies, single-chain fragments of antibodies, all are being looked at. Again, IgG is the most commonly, is the one on the market right now. The payloads are statins. VCMMA is used in Etcetras. On the lower right part, maytansine, that's TDM1, is used in Kadcyla. And then calicheamycin in the middle right, that's Mylotarg. So those are all drug linkers that are currently in approved drugs. Duocarmycin is another one that's been considered. There's a few others also, PBDs, perbenzodiazepines, that are also being currently in clinical studies. Conjugation strategies, you've already seen a little bit mentioned to this. I think it's Juan mentioned in his talk. You have surface lysine conjugation. An example of this on the market would be TDM1. Kadcyla, and you use various linkers to activate the surface lysines to attach the drug. Second approach would be interchain disulfides, and Etcetris is an example of this. And then genetic incorporation of single thiol groups that can be specifically conjugated by use of unnatural amino acids. So this is also currently being developed. A quick comment on the toxicities. The top three compounds, auristatins, maytansinoids, and PBDs, are examples of drug linkers that are being used. You'll notice on the far right, the operator exposure limit for 8 hours is in the nanogram level, with PBDs being currently the most potent. As a comparison to approved oncology agents, you see that those tend to be in the microgram range. So you're dealing with drug linkers that are considerably more potent, and of course, has been mentioned in some of the previous talks this morning in particular, the containment is a big issue when you're looking at this. So you're dealing with both the potency of your drug linker, where you need to protect the worker, and the necessity of aseptic handling of the antibody and the ADC to protect the antibody ADC from the worker. So you have both sides of the coin when you're making one of these. So I'd like to go over an example of the payload synthesis first, followed by an ADC conjugation. This is something that has been done at Carbage and AMSys. The sites will be mentioned here of our facility, and at the end of the talk, I'll give an overview of our company. But this is building a large molecule payload that was manufactured at our facilities. Building blocks being made under non-GMP, standard small molecule chemistry. 3 components, 11 steps, and 6 steps for each of those components, made in pretty much kilogram scale, followed by a 15-step solid phase synthesis in our Neuland facility in Switzerland. And this is under GMP now. So the building blocks non-GMP, subsequent steps GMP. We then have purification of the intermediate, followed by conversion to the payload in another site in Switzerland, followed by the conjugation, and then subsequently the fill finish activities at another facility. The final 3 balloons there are highly potent, and are also, the final 2 are then in clean room environments, increasing your cleanliness grades and GMP grade from grade D to grade C for the conjugation, and then from grade C to grade A for the fill finish activities. So you can imagine this is a fairly complex process. And the next slide, I just have an overview of how this is managed. So we have 40 chemical steps. We have multiple purifications in the synthetic as well as in the conjugation steps. You have manufacturing at 5 different sites in 3 different countries. 5 development teams involved in this. About a 2-year delivery timeline. 12 months work for scale up, and 1 project manager. A lot of interaction between technical teams and between technical teams, a project manager, and a customer. So this is an example of how complex these projects are. One of the things that also cannot be ignored, regardless of the company that's doing this, are the logistics between transferring material from one site to another. Or is it in another country, or is it intercontinental? All the paperwork that's involved with that is very important. And managing your lead times to be able to deliver the product to the customer on time. A quick talk about the characteristics or challenges of conjugation chemistry. So this is now dealing with the clean room activities. You have, as I mentioned before, both the environmental and health and safety activities or concerns, as well as the GMP concerns. Drug linker handling and aseptic processing, toxin dispensing and handling. You have your buffer preparations, which involves aseptic manufacturing, WFI, water for injection, not Wi-Fi, which I've referred to before in a previous talk. So your supplier should have Wi-Fi and WFI in case you're concerned about that. Conjugation. We had a previous talk about fixed, dedicated, or single use. What is the approach of the company you're going to work with? Your purification, type and scale. Do you know when you're going in, if you have a discovery-based process, do you already know how you're going to purify? Was the lab purification of your ADC done by centrifuge tubes, and now you're going into TFF or chromatography? That is a significant scale-up question. How is the bulk drug substance packaged? Is it done in a biosafety cabinet? Is it done under particle monitoring? Does your vendor know how to do that? Analytics. This is a crucial aspect of development that is frequently not taken as seriously as it should be. You have a mixture both of small molecule and large molecule, biomolecule analytics that have to be looked at. And again, environmental safety and health issues. And of course, there's always cost. So here's a very busy slide on analytics. I think just the take-home message of this is, if you look down the left side, you've got some pretty common methods, appearance and pH, for example, conductivity. You also have the more bio-oriented HIC, PLRP, mass spectrometry. Mass spectrometry of a small molecule is considerably different than of a protein or large molecule. Can your vendor do whole protein analysis, or do they have to outsource this? Again, you have reverse phase, size exclusion chromatography, and then you have the ion exchange, ICEF methods. So a vendor needs to be competent both on the small molecule analysis as well as on the antibody and the ADC side. Antigen binding ELISA, and then the more specific cell-based assays, residual host DNA, SPR analyses. Many vendors may actually have to outsource that. All these things go into your package for filing for an IND or for later on development and filings. And the regulatory approach has to be considered for your specifications when you're looking at release of your drug linker and release of your ADC and your antibody. All these things have to be considered. So how do you select a CMO? Of course, I represent a CMO, so I'm going to have a certain perspective, but I have worked on the other side. I've been on the buying side as well. So these are some of my experiences in working with a CMO in such a complex process. First of all, who owns the IP? This is not a trivial question. If you're the innovator, you should be the one who owns the IP, and you want to be able to keep it that way. Keep that in mind as you're selecting a CMO. What is the CMO's philosophy of project management? And what is their approach to process R&D? What is their regulatory overview? Things like that are all important. Factors including cost and speed. Don't forget that as an innovator or as a developer, you might have a timeline that may not mesh with the real world. So keep that in mind. The legal and quality related paperwork may actually prevent your starting on time. Something to remember. Some general considerations. So you have the most important aspect of this slide are the people in the ditch doing the actual work. Then you have tons of other people involved in making the process more compliant, shall we say. General considerations, again, reputation, corporate culture, how bureaucratic is the process on your side as well as on the CMO side. And what is finally, I think the final bullet point again, strong analytical QA as well as regulatory experience is very important. Some more specific considerations for an ADC. Again, you have a very complex supply chain. You have the issues, again, to re-emphasize safe handling of highly potent compounds. You would like to have a good supply chain for your payload as well as your antibody. Can the company do things other than antibodies, such as synthetic vaccines, analytical capabilities, the ability to work with external providers? So in other words, if you have other suppliers that you want to bring into this as a customer, will the CMO work with you on that? It's important. If the CMO requires exclusive supply of all the components, that limits your flexibility. And again, you notice a repeat emphasis on regulatory group. So just some things to think about when you're managing a project of this complexity. Re-emphasize, I mentioned previously, project timeline. Be sure to set realistic timelines for yourself and for the CMO. Be sure that you capture all the work. So this is an important, when you first formulate your request for proposal, do you know what all needs to be done? And can this then be captured in a project timeline? Have an agreement on the regulatory approach. Setting the start date may be very much dependent on your legal team and your quality teams. Tech transfer, work face-to-face. Very important, and I think if you Google project management pitfalls, you'll frequently find one of the pitfalls is if people don't have an interaction to build a relationship with the partner. Communication and avoiding project creep. So there's a lot of information here. I think the key point is communication and clear timelines that will help the project be, and also establish a system of dealing with issues when they go wrong. The more complex a project, the more likely there is that there might be challenges, unexpected results. If you're in an early phase, you might wind up having issues, scientific issues that weren't foreseen on a small scale. And how do you address that? Have that system in place before you get into the woods of the project. Take-home message. I hope I've shown you in a very brief time frame that an ADC is extremely complex. Both the toxin and the payload can be multi-step, highly complicated syntheses with many purifications. You have purification challenges both in the small molecule and in the ADC. I think we've seen that this afternoon as well. The analytical requirements cover both small molecule, protein, and conjugate. Don't forget your ES&H, environmental health and safety issues when you're selecting a CMO. And your project execution. Key to success, information, technology transfer, plan your timelines carefully, communicate. Select a CMO based on not only for your project, but also on your own culture. Kind of important. A little bit about this CMO. We're based in Switzerland. There's 3 red dots to the north and 1 to the south. The 3 red dots are in the German-speaking part of Switzerland, not too far from Basel in the west and Zurich in the east. And in the southern side is our Vienna facility, which is near Montreux. Our involvement with bioconjugates has been an evolution. We actually began highly potent API manufacturing in early 2000s. And during that time, actually, we're involved with a conjugation. And then our hypo activities accelerated and we became more focused. We've been involved with the toxin payloads of most or all of the areas in current ADCs. And more recently, we've become involved in the conjugation side with a build-out and qualification of a lab in 2014, where we do our conjugations now. We can do highly potent manufacturing in our sites, 3 sites in Switzerland, our bioconjugation in 1 site, and our sterile fill finish, final drug product, in Riom, France. And in that facility, we also can do lyophilization, which I've finally finished up with that. Just not to skip too much, our ability to do everything in-house, from conjugation, drug linker manufacture, and fill finish, all within the same company within Europe. And that is it. Thank you very much.